bio students. Today we are going to continue with 7.1 and we are going to focus on the topic of DNA replication. So once again, I'm using BioNinja because I like how it just simplifies the concepts and just really gets down to the nitty-gritty. And it just goes over the enzymes that you're responsible for knowing. So the first thing you should understand is that DNA replication is a semi-conservative process. That means that you are going to always retain one of the parental strands. So as this new DNA gets copied, you always retain one of the parental strands. So you have one parental and one new strand. Before going over the process of DNA replication, it's important for you to understand the structure of DNA as far as its anti-parallel nature. So what does that mean? That means that the parental strand here in blue, it's anti-parallel in nature. So one strand is running in a 3' to 5' direction, while the other strand is running in a 5' to 3' direction. So they're running in opposite directions. That's what anti-parallel means. All right, so let's look at the enzymes that are responsible to carry out DNA replication. And you're responsible for knowing the names of these enzymes and also knowing their function. So the first enzyme is helicase. So notice that helicase is right here, and helicase, what it does is separate the DNA. So it's got to separate it by breaking the hydrogen bonds that you find between the bases. The next enzyme that you're responsible for is DNA gyrase, which is also known as topoisomerase. You might see that name somewhere in your book. So DNA gyrase reduces the torsional strain created by the unwinding of DNA by helicase. So it helps unwind this DNA so that it doesn't just like twist back up like a rubber band. Okay. So the next enzyme you are responsible for are the single-stranded binding proteins. And what these proteins do is they help the DNA strands remain separated and also prevent the single-stranded DNA from being digested. So here we see the single-stranded binding proteins, and they help these two parental strands remain separate. So it stops the reannealing or the twisting process. All right, before we move on to the other enzymes, I just want to go a little bit into the definition of leading and lagging strands. So let's take some notes here. So something important to remember in DNA replication is that DNA replication always happens in a 5' to 3' direction. So here we see that this red strand is the new daughter strand being formed. So we see that it's always going to get copied in a 5' to 3' direction, right? Because it's anti-parallel to that parental strand. Now the other thing to notice here is that the fork is opening up in this direction, right? So notice that the leading strand right here, okay, is being copied in the same direction as the replication fork is opening up. So we call this strand the leading strand. We call it the leading strand for two reasons. One, because it's a continuous strand, right, that it's being synthesized in the same direction as the replication fork is opening up. So that's one thing to consider. The other strand that is being made is called the lagging strand, and the lagging strand is being copied in the opposite direction than the replication fork, okay? And that's because, remember what I told you initially, DNA replication always happens in a 5' to 3' direction. So if the parental strand here runs from 5' to 3', that means that DNA has no choice but to get copied in a 5' to 3' direction running opposite the replication fork. Okay? So that's the lagging strand, and is also known as the lagging strand because it's synthesized discontinuous. So that means that it's synthesized into these short little fragments here called Okazaki fragments. And these fragments here then need to be reannealed. Okay? So a little bit of information here about what the leading and the lagging strand is before we continue with the enzymes. All right, so continuing here. The next enzyme you are responsible for knowing is DNA primase. Now, the name changed recently. It used to be called RNA primase. And what DNA primase does, if we go up here, is that it makes a short RNA primer, which is about 10 to 50 nucleotides in length. And what the RNA primer does is it allows for an initiation point of DNA polymerase 3. So both the leading and the lagging strand are going to need a primer. Even though it's not shown here in the leading strand, it also needs a primer for this DNA polymerase 3. But once DNA primase makes the RNA primer, and notice that you know it's not RNA because here they color it in green, so we know that this primer is made of RNA. So once this primer is laid down, then DNA polymerase 3 can come in, locate the primer, and then it can start extending the new DNA strand, the new daughter strand. So it extends the chain. And the way that it does that is by adding nucleotides, right? Now, to be a little bit more specific, the way that DNA polymerase adds these nucleotides is that it adds the nucleotides in the form of DNTPs. So DNTP stands for deoxynucleoside triphosphate. So then what happens here is that out of these 3 phosphate groups, it uses 2 phosphate groups as the energy release to join the nucleotides in the growing chain of DNA. Okay? So that's the function of DNA polymerase 3, to add these DNTPs. All right, now again, it's not showing you here on the leading strand, but this DNA polymerase here is doing the same thing, right? It's adding the DNTPs. All right, so moving on. What does DNA polymerase 1 do? So I told you that in the lagging strand, we have multiple primers, right? So those RNA primers can't remain there. So DNA polymerase comes in and it removes these primers so that DNA polymerase 3 can then add the nucleotides. Same thing happened here at some point in the leading strand where there was an RNA primer. DNA polymerase 1 had to remove that primer at some point. Okay? So the last thing here is that remember I told you that this lagging strand is a discontinuous strand. So this lagging strand has been made in small little fragments called Okazaki fragments. So then these fragments need to be joined. So here's where another enzyme called DNA Ligase comes in, and what it actually does is that it joins the fragments. So a little bit more information about how it does it. It does this covalently by joining the sugar phosphate backbones together with a phosphodiester bond. And then you have a continuous strand here running in the opposite direction, of course. And it's no longer in the form of fragments. So then after these 2 strands have completely copied itself, you end up with a semi-conservative process of DNA replication.